Laboratory diagnosis of Toxoplasma gondii. Here we can use direct microscopy. Here we are detecting the tracheoid in the blood and the tissue cyst in the tissue biopsy. In the case of human, we can see the tracheoid and the tissue cyst and other stage of oocyst which are not present in the human. But in the case of cat, we can see all three stages: tracheoid, tissue cyst and the oocyst. But in the case of human, we can see the tracheoids and tissue cyst in blood or tissues biopsies. So here we can use the stains like Giemsa, PAS, silver stain or immuno peroxidase stain, or sometimes we can use the direct fluorescent antibody test. Here we are detecting the antigen, that means tracheoids in the sample. Other one is the indirect method, that means the antibody detection by using the Sabin-Feldman dye test, detection of IgG in serum by using ELISA or IFA, IgG avidity test, and detection of IgM in serum by using ELISA, or differential absorption test, detection of IgA method, and other thing, the detection of toxoplasma antigen by using ELISA. Other one, molecular diagnosis by using PCR or sequencing. Animal inoculation, here we are using the intraperitoneal inoculation into the mice. Tissue culture, peripheral macrophage cells line we can use, and the imaging method, CT scan or MRI scan, we can use to detect the Toxoplasma gondii. Next one, laboratory diagnosis of Butyraria bankrupti. In the case of Butyraria bankrupti, which is the common tissue nematode present in the Sri Lanka. Other one is the next one, Brugia malai. So we have to differentiate these two, the Butyraria and Brugia malai. So the first laboratory detection is demonstration of microfilaria. So we can use directly, we can use the blood smear, that means direct wet mount, that means take a drop of blood, and when we observe under the microscope under 40, we can see the movement or motility of the microfilaria. Or sometimes we can use the stain smear. So here we have to use thick blood film, and before staining we have to dehemoglobinize, that means remove the or destroy the red cells, and then we have to go for the staining. We can use the stain, Giemsa stain or Delafin stain to detect the microfilaria. Then next one is the antigen detection by using ELISA or ICT. Other one, antibody detection, we can use ELISA. Other one is the imaging method by using X-ray. Molecular method we can use PCR. Synodiagnosis we can use. Synodiagnosis means by using the vector we can inoculate the suspected sample. So if any parasitical stage is present, we can see the parasitical stage in the vector by dissecting the vector. Other method, that means indirect method, is eosinophil count high. Normally in the case of parasitological infection, the eosinophil count is very high, and cellular assays we can use. So now we will discuss the microfilaria of the Butyraria bankrupti and Brugia malai. So we have to differentiate these microfilaria. So when we go through the morphological features, the appearance, normally the Butyraria bankrupti graceful or sweeping curve, that means a primary curve only we can see, that means very single curve, that means less coiled. Size is compared with the Brugia malai larger. When we consider the cephalic space, that means anterior part, in the case of Buchereria bangrofti, length to width ratio 1:1. Then nuclei, body nuclei, we call body nuclei. This is larger, size is same size, and the well separated and evenly distributed in the body. Then when we look at the tail part is the pointed tips, and there is no tail nuclei, and the excretory pore is not prominent, that means very smaller. In the case of Brugia malai, when we consider the appearance of the Brugia malai, it is crinkled with secondary curve, that means overcoiled. When we compare the Buchereria, Brugia is overcoiled, that means secondary curve we can see. Then when we consider the size of the Brugia malai, the size is smaller compared with Buchereria bangrofti. When we consider the cephalic space, which is larger, that means length to width ratio is 2:1, but in the case of Buchereria bangrofti, 1:1. When we consider the body nuclei, in here the Brugia malai, it is larger and overlapping, and the size of the body nuclei is vary. And when we consider the tail part is pointed, and we can see the tail nuclei, 1 or 2 tail nuclei in the Brugia malai species. The next one is the excretory pore is prominent, that means larger excretory pore we can see in the Brugia malai. Another thing, in both cases, Buchereria and Brugia, we can see in the sheath, but in the case of Brugia malai, the sheath is pinkish color, but in the case of Buchereria bangrofti, color is whitish or colorless, or very light pale purple color. So here we can see the typical morphology of the microfilaria. So in the case of Buchereria and Brugia, which are present in Sri Lanka, so in the case of microfilaria, in both cases Buchereria and Brugia, we can see in the sheath, and the cephalic space is the anterior parts. We call actually the head part or mouth part we call cephalic space. So here we have to mention this cephalic space. We cannot label head part or the mouth part. This is the specific name for the microfilaria of this tissue nematodes. So here according to the length and the width ratio, we can differentiate the Buchereria and Brugia. The length to width ratio 1:1 in Buchereria bangrofti. In the case of Brugia malai, length to width 2:1, that means length is 2 times and width is 1 time. Then when we consider the body nuclei, in the case of Buchereria bangrofti, evenly distributed, same size, and they separated, but in Brugia malai, not evenly distributed and overlapping, and the nuclear size is differ. And 1/3 of the anterior part we can see in the nerve ring, and the next one, the posterior part, that means tail part, it is pointed, and in the case of Buchereria bangrofti, we cannot see the tail nuclei, but in the case of Brugia malai, we can see the 1 or 2 tail nuclei. So here we can clearly differentiate Brugia malai and Buchereria. Here we can see the first picture, very darkish color. We cannot see the separated nuclei, and the anterior part we can see very longer, very lengthy cephalic space. and here very difficult to identify the tail nuclei, but the body nuclei we can see in very darkish color. And in the case of Butyraria bangrafti, very clearly we can see in the separated body nuclei. And the posterior part we cannot see the tail nuclei. So here also the morphology, the appearance, when we look at the appearance of the microfilaria, first one, the upper picture is the Burugia malai, down picture Butyraria bangrafti. You can see the curve, the only single curve in Butyraria bangrafti. In the case of Burugia malai, upper picture, it is over coiled, that means too much coiled. And here you can see the sheath is very pinkish color in Burugia malai, but in the case of Butyraria bangrafti, not like that, very pale color, purplish color. So here you can see the appearance, the single or less coiled or curve in Butyraria bangrafti, but Burugia malai very higher secondary or more curve. But size, when we consider the size, Butyraria bangrafti is larger, Burugia malai is the smaller one. And here you can see the tail part, you can see the tail nuclei, and the body nuclei. When we consider the body nuclei, we can see in Butyraria bangrafti very separated, evenly distributed, same size. But in the case of Burugia malai, not like that, unevenly distributed, the size may vary and the overlapping. Then we will move to laboratory diagnosis of malaria. So here microscopic testing, peripheral blood smear we can use. This is the gold standard method. Here we have to prepare thick and thin smear. So we can prepare thick smear separately or thin smear separately, or in both thick and thin in the same slide. So why we prepare thick film? This is more sensitivity, that means increase the identification, the detection capacity. That's mean we are adding more than one drop of the blood, so increase the chances of detection. But in the case of thin blood smear, we used to identify the species. Why we cannot detect the species in thick blood film? Here thick film, after preparation we have to air dry fully, it should be dry. Then we have to go for the dehemoglobinization. In dehemoglobinization, what happening? The RBC, normally in our blood RBC the number is very higher. So without removing the RBC, when we are doing the staining step, the RBC get stained, so we cannot identify the microfilaria. That means it's overlapping. Microfilaria and RBC also get the same color, so we cannot differentiate the microfilaria. So before staining we have to remove the RBC, so we have to do the dehemoglobinization by using distilled water. That means remove the RBC. So the other thing there, for the species identification we should know the morphology of the RBC. So we already discussed that means the RBC. In the case of Plasmodium vivax, the RBC morphology is enlarged and the pale color. In the case of Plasmodium ovale, the RBC enlarged, oval shape, sometimes fumigated. But in the case of Plasmodium falciparum and the Plasmodium malariae, the red cell size is same to the normal red cell size, and the color is little bit pale compared with the normal RBC. So next we will go for the fluorescent microscopy. Then we can do the quantitative buffy coat examination. Here we are detecting the antigen, that means the parasitical stages. Non-microscopic tests are antigen detection test by using ICT. The one antibody detection by using ELISA, and we can do the culture and molecular study by using PCR. And thank you.